Hello, everyone, and welcome to today's broadcast, Understanding Cancer Through Whole Genome Screening. It is presented by Andrew Ravanelli, Ph.D., Senior R&D Scientist for Genome and Epigenome Editing at MilliporeSigma. I am Judy O'Rourke-Laverts, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by MilliporeSigma. For more information about our sponsor, visit www.milliporesigma.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Ravanelli. I will now turn the presentation over to him.
All right, thank you for joining this webinar. My name is Andy Ravanelli, and I'll be discussing functional genomic screening approaches to discovering genes and pathways that are relevant to cancer and to other cellular phenotypes. So the life science business of Merck KGaA in Darmstadt, Germany operates as MilliporeSigma in the US and Canada, and I'm a senior R&D scientist with MilliporeSigma in the Genome Engineering Division. So cancer is, of course, the uncontrolled and unregulated growth of cells which metastasize throughout the body. There are a number of capabilities acquired during the multi-step development of human tumors that promotes cancerous behavior. Cancer can be considered a genome disease at the cellular level, and the sequencing and mapping of the human genome has really paved the way for cancer genomics and is being used to define molecular targets for tumor-specific properties. Remarkable progress has been made regarding mechanism-based targeted therapies and has really been informed by these hallmark characteristics, a handful of examples of which are presented here. New therapeutics are often designed against specific molecular targets that enable one or more of these capabilities in cells. So in principle, these targeted approaches have relatively fewer off-target effects and thus non-specific toxicity. However, in reality, positive clinical outcomes really are often transitory and followed by relapses and tumor resurgence. So this may be due to some redundancy in signaling pathways or because tumors may shift their dependence between hallmark abilities as they evolve. And so a powerful strategy is then to take a functional genomics approach to develop a comprehensive understanding of the genes and pathways that direct the adoption of tumorigenic cellular behaviors such that more effective long-term approaches can be developed and utilized. So what is functional genomics then? Functional genomics is a field of study that examines the relationships between genes and complex cellular processes on a genome-wide scale. From a foundation of genome sequencing and high-throughput analysis, it can involve things like mutagenesis, targeted gene knockouts and knock-ins, activation and repression of transcription, transgenic overexpression of genes, or disruption of protein translation. The most comprehensive use of these approaches are in genome scale screens that allow for discovery of relevant gene function. So the ease at which one can use functional genomic tools to target many genes at once has really opened up the possibilities and the accessibility for whole genome screening. We now offer or often rely on short non-coding RNAs that can be transfected or in the case of CRISPR, expressed along with Cas9 or transduced in viral formats for delivery. Genetic screens are unbiased and they allow for discovery of drug and disease relevant pathways and phenotypes that were not previously considered. Loss of function and gain of function screens are complementary to each other, and screens are very high throughput and they can yield whole genome resolution. Screens are efficient and accessible ways to dissect and understand gene function. Targeting tools are designed strategically for optimal targeting to increase confidence in your phenotypic, and they really require much less time and cost than protein-based screens like zinc finger nucleases and TALENs, for example. So Sigma Aldrich, and now MilliporeSigma, has a long history of expertise in functional genomics screening tools. Starting over a decade ago with our Whole-Genome shRNA libraries, our portfolio has grown, often through collaborations with leaders like the Broad Institute and the Wellcome Trust Sanger Center. And we were one of the very first to offer CRISPR reagents and received multiple awards over the years for our innovations and offerings in CRISPR epigenetics and CRISPR screening tools. There are two primary types of screens for which we offer support and that are useful for functional genomic analysis. Pooled screens, which combine thousands of clones into a single tube, and arrayed screens, which consist of thousands of unique wells of individual clones. Pooled screens allow for rapid identification of relevant target genes, while arrayed screens are perfect for high-content imaging and cellular phenotyping on a fine scale. Pooled screens are generally the most widely used and are pretty straightforward and upfront, but they require much more of a time and expense investment on the back end during data deconvolution and follow-ups. Whereas arrayed screens require a much more substantial upfront investment of labor, but can yield a much broader diversity of data points and much less back-end effort because all of the data has really already been collected. So what do some of these libraries look like? The first category contains knockout gene editing tools, and we offer both arrayed and pooled library approaches. The Sanger Arrayed CRISPR library, made in conjunction with the Wellcome Trust Sanger Center, contains CRISPR guide RNAs for over 17,000 genes with 2 individually arrayed guides for each gene. The guide plasmid contains a blue fluorescent protein cassette, which is useful for visualizing transduction or transfection efficiency of the library. And in addition to the whole genome arrayed library, we've also developed smaller panels derived using up-to-date, expert-curated CRISPR databases. Instead of relying on a single individual or a single source for gene family lists, we've combined and compiled and cross-referenced numerous resources, including the CAG database, gene ontology sets, the druggable genome database from experts like WashU, and our own manual curation of PubMed IDs. So this is an ongoing exercise that allows us to create non-redundant, research-focused gene panels, a variety of cellular functions and categories, which includes 5 cancer-related gene panels. Importantly for this, these small panels can be customized to include or exclude any number of genes, or you can design fully bespoke panels of your own gene sets. And for those budget-minded, these focused panels allow for targeted arrayed screening at a lower cost. So the real power of arrayed screening is in the ability to incorporate high-content imaging and analysis across a range of metrics, and a handful of examples are listed here. For this, cells are plated in 96 well plates, and they're treated with the clone library, followed by either selection or phenotyping. Wells are then analyzed individually and assayed for features like cell death and proliferation, cell morphology, or reporter fluorescence. Because each well is assessed individually and can include multicolor imaging, mixed populations of cells can be analyzed within a single well. This becomes incredibly useful when considering the heterogeneity within the tumor microenvironment. Tumor cells are often mixed with cells of normal function to promote tumor growth and tumor maintenance over time. And then individual gene function can then be assessed to dissect particular cell-cell interactions within this arrayed screen. High-content imaging also allows for more fine-scale image analysis using, for example, subcellular protein localization. So in this example, STAT1 and STAT3 proteins have been fused to GFP and RFP respectively, revealing cytoplasmic localization of these proteins on the top under normal conditions. But then upon stimulation with type 2 interferon, for example, STAT proteins then translocate to the nucleus. And you can see nuclear localization of this staining, or of this GFP and RFP fluorescence. Software detection of cytoplasmic versus nuclear staining then elevates the utility of this system to allow for high-content image analysis of protein localization in response to disrupted gene function, for example, using the Sanger arrayed library. Knockout of STAT3 here using the Sanger arrayed CRISPR guides results in a significant decrease in RFP positive cells, as you can see in the middle. And this figure also shows the usefulness of having the BFP incorporated into the guide plasmid to ensure that you've achieved effective delivery and that these guides are capable of then disrupting gene function in cells that have taken up this cassette. So in addition to the arrayed libraries, we offer both the Gecko CRISPR pool that was developed by the Broad Institute, as well as our own Sigma CRISPR pool. And these libraries have great utility in knockout screening in cell culture and in vivo models, as evidenced by this really cool paper from Phil Sharp's lab, identifying critical genes driving tumorigenesis and various tumor subtypes upon injection of cells that were treated with the Gecko whole genome CRISPR library into immunocompromised mice. So although knockout technologies have been established and adopted across multiple platforms, there's really been a lot of new excitement lately in the development of tools that regulate gene function independent of changing the DNA. CRISPR activation and CRISPR inhibition, for example, offer all of the sequence-specific targeting of the CRISPR complex to deliver effector domains that regulate transcription of genes. We recently launched a set of new CRISPR activator tools. So why add CRISPR activators and CRISPR screening then to your toolbox? CRISPR-A is complementary to existing loss-of-function technologies, as each of these enriches for different sets of genes responsible for the same phenotype. Unlike loss-of-function technologies, CRISPR-A is fairly unique for gain-of-function screening. And because it's nuclease independent and the design rules are fairly strict, the risk of off-targeting effects are substantially lower with CRISPR-A. With CRISPR activators, there's really little to no risk of genome instability from double-stranded breaks, again, because there's no cutting. And when performing knockout screening using a cancer cell line, sometimes you have to worry about the ability to completely knock out high copy number genes. And this really isn't a concern for activation screens. And by activating transcription at the endogenous locus, instead of transgenic overexpression of a gene, for example, of a defined isoform, CRISPR-A allows the cell to dictate isoform expression and splice variation. So George Church's lab published a nice comparison paper of the different CRISPR activator technologies and showed CRISPR-Sam to be the strongest transcriptional activator system. That's why we chose to optimize and produce the CRISPR-Sam lentiviral whole genome libraries. So the CRISPR Synergistic Activation Mediator, or SAM, was described by Feng Zhang's group at the Broad Institute and is a three-part CRISPR-based transcriptional activator complex. At its core is the DCAS9 VP64 protein that forms the basic synthetic transcription factor, and it complexes with a guide RNA to form the RNP targeting complex. But the stem and tetraloops of the guide scaffold in this context have been modified to contain RNA aptamers that bind and recruit additional transcriptional co-activators to the complex to help maximize transcriptional output. We found that the original vectors could benefit from some functional lenti-titer optimization. So our new modified vector backbone produces substantially higher levels of functional virus so that our new functional titer for SAM meets the efficiency that's required for whole genome screening with a pooled library of guides to achieve the desired levels of coverage. So the CRISPR-Sam library follows a fairly universal guide design rule, improving guide design efficiency. The library was validated using a whole genome screen for BRAF inhibitor resistance in A375 cells. BRAF is the most potent activator of the MAP kinase pathway, and this pathway is mutated in an estimated 30% of all cancers, with mutations in BRAF specifically found in approximately 7% of all cancers. And nearly all of the observed mutations in BRAF are the same as the one harbored by these A375 BRAF mutant melanoma cells, resulting in constitutive activation of the pathway. These cells are integrated with the SAM helper components and then transduced with the guide library. The BRAF inhibitor, PLX4720, was used to enrich for resistant cells that escape selection. Genomic DNA is then harvested, and guide representation was established by next-generation sequencing analysis. So during our initial validation of the library, we showed high guide representation across the library for a number of different cell lines and screening approaches. And as you can see in the graph above, for the BRAF inhibitor screen, we showed strong enrichment of a cohort of guides that are circled in blue following BRAF inhibitor selection. So our screen produced EGFR as our top hit on the right by magic analysis, matching the top hit from earlier studies from Feng Zhang's lab and others that EGFR is critical for mediating this pathway and this resistance. We also pulled out BRAF, or mutant BRAF in this case, validating another study showing that overexpression of this gene can overcome drug inhibition. So top hits from the Konerman SAM screen from Feng Zhang's group were then validated using individual guides for improved survival under selection and showed increased mRNA and protein resulting from transcriptional activation analyzed by qPCR and Western blots. In this schematic of the pathways implicated in proliferation and survival of these cancers, we can see the established models of reactivation of the RAS-ERK pathway and the parallel pathway of PI3 kinase AKT signaling. The top hits from the screen are highlighted in blue lettering, where those previously validated using an ORF overexpression library approach have been underlined in green. So importantly, a number of novel genes are now implicated in BRAF inhibitor resistance from this, including a family of ITG receptors not previously described. So this screen really shows the importance of using complementary approaches to identifying relevant genes and pathways to your phenotype of interest, because they can reveal distinct sets of genes and interactions that might otherwise remain undiscovered and that may open up new avenues then for therapeutic exploration. So finally, I'd like to discuss a tool separate of CRISPR using an established mechanism of gene repression and translation inhibition of RNAi. So we offer a number of shRNA screening platforms, both pooled and arrayed, for whole genome, focused, and custom libraries. For this, I'd like to highlight an exciting paper from earlier this year that provides a comprehensive look at functional genomic discovery from a forward genetic screen to clinical relevance. And this study from the Gomes lab combined an experimental mouse model for tumor metastasis with a genome-wide screen to ask what genes regulate dormancy in these disseminated tumor cells. So they injected dormant bone metastatic, or DBM, these are luminal ER breast cancer-derived cells, into immunocompromised mice. The goal of this screen was to probe the genetic basis for several characteristics of these cells. First, these injected cells disseminate and subsequently undergo homing to the bone tissue. They next undergo a period of dormancy or latency, whereby no resulting lesions or secondary metastases form for many months in these mice, similar to how they function in humans for many, many years or decades. Finally, upon some uncharacterized stimulus, they exit the latent phase and lead to symptomatic lesions and metastases. So for the screen, they applied the whole genome mission shRNA library to DBM cells in 10 subpools and sequenced the cells for representation. The 10 subpools were subsequently injected into the ventricles of 10 mice each. And after 4 months, the resultant lesions were harvested and sequenced to reveal shRNA enrichment. So genes that were known to regulate proliferation and apoptosis were excluded from this analysis, and they were left with a list of about 320 genes. From there, only 2 of the candidates were found in at least 2 lesions and with at least 2 independent shRNAs. And the authors chose to follow up on one of these for exploration, and that's the gene that encodes MSK1. So the MSK1 shRNAs were then validated by Western blot for protein levels and by homing assay to the bone, and further validated using CRISPR knockout on the right, showing a similar response. These cells were then transplanted into mice and assessed for symptomatic metastases. MSK1 knockdown correlates with decreased bone metastasis-free survival. In addition to a deeper dive into the molecular mechanism of MSK1 activity, which I'm not covering here, and the authors really went comprehensive on this, they examined bone biopsies from patients with ER-positive breast cancer and found that bone metastasis-free survival positively correlated with high levels of MSK1, so that they then could potentially stratify patients for potential prognosis. And they concluded that MSK1 regulates expression of differentiation genes in luminal breast tissue, and that in the absence of MSK1, differentiation programs are compromised and can lead to metastatic progression of these cells. So this paper and studies like it really exemplify how the utility of unbiased forward genetic screens and functional genomic analysis can reveal novel gene pathways and functions that have direct relevance to clinical outcomes for patients with cancer. And they can provide an excellent starting point for the development of novel drugs and therapeutic approaches to improve development pipelines. So although whole genome screening is pretty straightforward and shouldn't be scary if you use our kits and reagents, there are a number of considerations to explore before you start any screen. Screens take reagents and they take manpower and they really take time. And you need to know the ins and outs of your cell line or your approach as it pertains to proliferation rates, karyotype, drug resistance, and transduction efficiency, for example. You need to understand the effect of your drug on your cells. For example, how long do you need to screen with your drug to get a good enrichment or phenotype? And you need to have clear expectations for your phenotypic outcome so that you're going at this at a really strategic and targeted manner. Next generation sequencing requires expertise in the use of specialized equipment or access to it, and in bioinformatics support. And you need to try to anticipate how you'll go about validating your top hits and following up on your screen. These are all pretty standard, but they're things that you're really going to want to include in your planning before you start a screen. So we've covered a lot of ground, so I'll just finish with a summary of the options that you have when looking for functional genomic screening tools. We have an extensive portfolio of gene editing and expression reagents that includes lentivirus and DNA and RNA formats, along with synthetic oligos and RNAs and proteins for whatever suits your individual needs. There are a number of library options, from pools to arrays, both off-the-shelf and custom panels of a variety of sizes. And we offer formats for knockdown and knockout, and activation and overexpression screening, which can even be performed in parallel or complementarily to one another. And just as important, we have the historical and the technical expertise to support you and your projects all along the way. And I really hope that you'll reach out to us if you have any questions about how we could possibly help you with your next CRISPR or genome engineering or functional genomics project. So I hope that you come away from this excited to explore screening approaches to enhance your research pipelines. And I'd really love to try to answer any questions that you might have in the remaining time. But if for some reason we get cut off, please feel free to email me at andrew.ravanelli@sial.com or at MilliporeSigma.com. And thank you for listening.
Thank you, Dr. Ravanelli, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If If you have questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, would you expect a CRISPR knockout or SAM activator screen to be able to discover the MSK1 target?
So would we be able to find MSK1 with either a knockout or an activation screen? That ultimately depends on the context. If you're doing the same screen, I wouldn't necessarily, if you're doing the exact same screening approach using the same drug, I wouldn't necessarily expect to find the same genes in both a knockout screen and an activator screen. But you could, using these two screens, you could approach the same signaling pathway or the same protein interactions or gene interactions from two different angles and identify different pathway members along the way. But because the targets and the guides that target each of these genes, like MSK1, are present in both a knockout or an shRNA or a CRISPR activator library, you would expect to find that gene in screening depending on the context of your screen.
Thank you. Next one is, you show 1,000-fold activation with your SAM library. What is the maximum level of activation the system can achieve?
So that's a great question. And it's another kind of hedging answer. The SAM activators, been one of my favorite projects to work on. And so the most important thing, and really what the power of the CRISPR SAM activator system is that because you're regulating gene function from the endogenous locus, the context in which you are trying to achieve activation or inhibition is very much dependent upon the cells and the current physiology of whatever cells or cell line you're using. So the CRISPR SAM system activates at the endogenous locus. If that locus is already highly active and expressed in your cell line, you can't recruit more transcriptional machinery to that locus. So if the gene is already expressed at very high levels, you're not going to see much, if any, activation. You will see much, much higher activation the lower the gene is expressed. And you can avoid, unlike transgenic overexpression, you can avoid toxic overexpression of genes with this system because, again, we're using the cell's endogenous transcriptional machinery at the cell's locus in the genome. So it depends on the context.
Thank you. Is one approach more powerful than another? For instance, is RAID or pooled screening more effective in finding candidate genes?
I think I'll have the same answer for pretty much every single question, and that's context dependent. But pooled screening, it depends on what you mean by power. And a RAID screening and pooled screening should be able to, if you're using these with the same context of a single drug or a single phenotype, should be able to pull out a similar list of genes, although genes with genes that give more subtle cellular phenotypes may fall out in a pooled screen, whereas those may be able to be brought to the forefront in a RAID screen because you can see every single well assessed individually. So you can see all of the genes that are responsible for a particular phenotype in an arrayed screen, whereas in a pooled approach, you'll see the top hits that will come out, or the most important, or the genes that are responsible for the strongest enrichment. And from those, in either of the screening contexts, you would choose which hits to follow up on for further validation and continued study. So neither one of them is more powerful than any other one necessarily. It just depends on what approach you are more interested in, how many guides you can follow up in, and what type of screening approach and what type of data you want to get. If you want to get detailed phenotypic data or fluorescent data or protein localization, you need to use an arrayed screen. If your system is high throughput, looking for cell survival or cell death, a pooled approach is definitely a fast way to get to that information. Yes.
Thank you. Okay. And last one is, are biological replicates required for pooled screening?
Yes, that's a simple answer. Biological and technical replicates from a scientific or from an experimental perspective are always required to achieve statistical significance. If you want statistical significance, if this is just a pilot and you're looking for a candidate list of genes to follow up on, as you would pretty much with any screen, then you don't necessarily need biological replicates, but for maximizing your confidence in what your top hits are, you'll always want to have biological replicates. Screens are always going to be hypothesis generators or broad approaches to give you a list of genes to follow up on. They will never give you the answer to your question. They will never give you all of the phenotypic data that you need to address gene function or functional genomics. Screens are always the first step toward a more in-depth and thoughtful and comprehensive study. And so for that, biological replicates are excellent and necessary for getting statistical significance, but you're definitely going to follow up with much more comprehensive research programs for your top hits when you get done.
Well, thank you. I would like to once again thank Dr. Ravanelli for his presentation. I would also like to thank MilliporeSigma for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11th, 2019. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Bye.
Thank you very much.